So the question is then, how can the binding of a growth factor at the receptor lead to changes across the membrane and down into the cytosol? And how can the dysregulation of these programs lead to cell transformation? So a big change in phenotype, such as differentiation or apoptosis or proliferation. So these small signals at the plasma membrane are driving these big changes in cell signaling. And early on in research, people knew RAS was an important molecule that was involved in cancer, and they was wondering, well, does RAS play a role in normal cell signaling to drive normal cell processes? So the idea was, if you can understand normal cell growth and normal cell signaling, you can then understand aberrant cell growth. And then you can look at the role of these oncogenes and tumor suppressors to see how they affect cell growth. So typically when we talk about a signaling pathway, we talk about something that reaches from the cell surface, from an external signal, all the way through to turning on gene expression in the nucleus. So to understand this process, people have been doing studies in normal cell fibroblasts. So these are normal cells which you can grow in tissue culture, and you can study replicative cell division and growth. And one way to do this is to get your normal fibroblasts and serum starve them. So you restrict nutrients and growth factors from these cells, and then you push the cells into a quiescent, a non-growing state called G0. And last week we looked at G0 and then the cell cycle with G1 S phase, G2 mitosis, and we looked at cell growth. So in the lab, you can get these fibroblasts, you can serum starve them, and then you can add fresh serum, and this adds all of these unknown growth factors, these uncharacterized growth factors, to cells, and it starts them growing synchronously, and they go into cell division at the same time. And you can study this process, and the question was, could this be similar to the way that you take normal cells and you trigger them into growth in a cancerous state? So the idea was, if you can understand normal cellular pathways and normal cell growth, then you can look at aberrant cell signaling and the role it plays in cancer. So given that we're thinking about cancer as a disease of uncontrolled cell proliferation, we can then study how cells grow. So both cancer cells and normal cells utilize the same machinery, the same signaling pathways, and in cancer cells, these pathways have been disturbed. So that's the hypothesis. And in modern genetics, we can study the response of these synchronized fibroblasts, these cells, in response to a growth factor. And then when you add the growth factor to the cells, you can look at which cells are turned on within that cell population. And the cells will grow in an ordered way, so that when you take samples from the cells, all the cells have just entered G1, or all the cells are just entering S phase, and all the cells are just exiting into G2. So you can study progression around the cell cycle, because you can serum starve the cells, and then you can add fresh serum and all the growth factors to get cells growing synchronously. So when you take that technique and then you study cell samples from these cells using microarray experiments, you can actually look at gene expression across the genome. So you can look at all genes that are turned on after serum addition. And that's what this slide, this diagram here, is showing. It's showing a gene expression profile at time 0 when you add some growth factors, some serum to cells that are in G0, and then you push all the cells to start growing. And then you take other samples 30 minutes after the serum was added, and then you take other samples at 60 minutes and 120 minutes, so 2 hours. And the genes that have been turned on immediately after serum addition, it was thought, well, maybe these are the genes that are involved in controlling cell growth, and therefore this is going to tell us which genes may be involved in cancers. So these were the experiments they did. They triggered cell growth in a synchronized cell population, took samples after various time frames, and they looked at the sets of genes that were turned on and off. And even if you just look at this, this heat map here, you can see straight away that at time 0, there's a bunch of genes that are not expressed at any great extent, but within 30 minutes, these are all turned on because they're all bright red. So if you look down here, high gene expression is red, low gene expression is blue. So these were lowly expressed, and the addition of serum led to a whole bunch of genes being turned on. Okay, and likewise, there's a bunch of genes that were on, and that serum addition caused them to be turned off. Okay, now these genes are turned on immediately, whereas if you look down here, there's a bunch of genes which aren't turned on immediately, but they're turned on after a certain delay. Okay, so there's clearly some cell signaling going on here at different times, and I'll try and give you an appreciation of when these are turned on and how they're turned on by looking at a diagram of the cell in relation to this data. So what we have now are groups of genes that are expressed at different times. And another subtlety of this experiment is that you can add an inhibitor of protein synthesis, a chemical called cyclohexamide, to the cells, and this will inhibit protein synthesis, which says that these genes are turned on by existing proteins that exist in the cell. So the signaling machinery that's in place to activate these genes must have been in place beforehand. Okay, so when you add cell signaling molecule to a cell, a growth factor, the initial changes in the cytoplasm occur by switching on proteins that are already in place. And these pathways that are already in place then signal to the nucleus to turn these genes on. Okay, so this happens in the absence of new protein synthesis, indicating that the machinery must be in place for the process to occur. So when you look at some of these genes that are turned on within 30 minutes of adding a growth factor, then you're seeing genes such as FOS and JUN, which are important oncoproteins, which are important in turning on a bunch of genes, and there's a whole bunch of genes, and they're located throughout the cell, and they have important roles in driving growth and these phenotypic changes to cells. So what we're seeing here is we're adding a growth factor to these cells, these synchronized cells, and immediately we're able to switch on some sort of signaling pathway because proteins are already there, these things are already in place, and they're activated straight away. So so within 30 minutes of adding the serum, the growth signal, we've activated signaling pathways that have gone from the plasma membrane through to the nucleus. And what you see here is gene expression. So we've managed to turn on a whole bunch of genes within a short period of time from the addition of a growth factor. So we're going to look at how this signaling pathway, we'll look at an example of one of these signaling pathways. And And then what we see later on is a bunch of genes that are turned on after another maybe hour and a half. So there's a longer process before these are turned on. And the way you can rationalize these bunch of genes is by looking at the genes that are turned on. These genes, express the genes are transcribed into messenger RNAs, and then these messenger RNAs are translated into new proteins. And then some of those new proteins are actually transcription factors which go back into the nucleus to turn on a bunch of delayed genes. So these genes here require new gene synthesis. So the cyclohexamide would block that particular pathway because it blocks new gene expression, but this pathway is not blocked, so that signaling pathway is in place. So that tells you that these delayed genes requires new gene synthesis, and new transcription factors are made to turn on a second bunch of genes. So you get an idea of the cellular processes that are going on after the addition of growth factors to cells. And then we're going to look at one of these signaling pathways. We're just going to build upon our previous knowledge to put together one of these signaling pathways. And then we'll look at some of these are some of the key oncogenes and tumor suppressor proteins are involved in these pathways. So effectively, the idea that genes involved in normal cell growth, when perturbed, are involved in cancers proved to be a very valid hypothesis. And these immediate and early genes that are expressed are often found to be expressed at high levels in cancer cells. And therefore, an understanding of signaling processes will help us understand cancer.